Hi everyone and welcome to Lecture 10. Today we're going to go through a variety of different infections that are spread person to person. And keep in mind this is just a sample, that there are many more that we do not have time to discuss because they're basically infinite numbers. So the first system we're going to look at is the respiratory system. And this is an important one because think of it, you always have to breathe, and it's something that you're not even conscious of. So every time that you're breathing, you may be susceptible to picking up certain microbes around you. So as I was just saying, respiratory is the most accessible portal of entry because, again, you have to breathe. And it is constant all day, every day, whether you're awake, whether you're asleep, you are always breathing, which means you are always able to take in microbes through this way. Now, the first thing that I mention on this bullet point is the idea that the respiratory infections are usually bacterial or viral. Fungal you usually only see in immunocompromised people. And when we talk about respiratory infections, you have to keep in mind that there's the upper respiratory tract and there's the lower respiratory tract. And the type of infections you'll get, the severity, they differ depending on which one of these areas is being affected. So when you think of the upper respiratory system, what do you think of? What's part of your upper respiratory system? You have your nose, you have your mouth, and you have your trachea. Okay? These things in the upper respiratory system, they are constantly exposed to pathogens because, again, like I was saying, the idea of breathing, and if you think about the mouth is part of both respiratory and digestive. So that gets hit double in terms of taking in pathogens. Now, the good part, I shouldn't say really good part since you do get infections, but the good part about upper respiratory infections is they're usually not as serious. And we'll go through some examples in a minute and why they're not as serious. Then you have the lower respiratory system. And when you think of the lower respiratory tract, that's now deep within your body, your bronchi and your lungs. Okay, and the bronchi are those branched ends of the trachea, so you have one for each lung. Now, your lower respiratory tract is supposed to stay sterile, okay? If it does get infected, then you have pneumonia. And when you think of what pneumonia is, that's inflammation of the lungs. And you have to be very careful working in hospitals because your patients will be very susceptible to pneumonia. Okay, especially the older and immunocompromised patients. Now, once a respiratory infection is lower, it's going to be much more dangerous for the person who has it. And we'll give some examples of that. But just keep in mind, first of all, the respiratory tract is the most accessible portal of entry. Okay, you are always breathing. And keep in mind the two different tracks. What exactly is making them up? And the idea that upper respiratory infections are not going to be as serious. They're not going to show as severe symptoms. Okay, so we're going to go through some examples of exact infections that you or your patient might get. The first set of infections that we're going to look at, these are some bacterial diseases of the upper respiratory system. So keep in mind, these are all caused by bacteria, and these are the upper respiratory system. Now, interestingly enough, the middle ear is actually considered part of this because if you think of any time you have ear infections, your sinuses can be infected and it affects the whole upper region of your body. Now, the ones that I want to point out on this slide, first up we have pharyngitis. Okay, pharyngitis is a fancy way of saying strep throat. Okay, classic sore throat. And as we saw in lab, this infection is caused by strep pyogenes. Now, funny enough, the next one on this list, scarlet fever, is also caused by strep pyogenes, But now what's different from pharyngitis, the reason why symptoms will look more severe, is that exotoxins are now circulating. Then the last one on this list is diphtheria. And with diphtheria, I want you to write down DI for diphtheria and DI for die. Okay, diphtheria is the only upper respiratory system infection that we're going to talk about that is life-threatening. Okay, so out of the upper respiratory system infections, diphtheria, always think die, life-threatening. The reason this one's so problematic is that the patient will have exotoxins circulating in their blood, and that's going to end up damaging the heart and the kidneys, and you can end up with toxemia. So now you have these toxins circulating all over, which can give septic conditions, and your patient can very, very quickly lose function of their organs. And once a patient's kidneys fail, that usually means that they're not going to make it. Okay. Now the good part about this is that diphtheria is part of the DTaP vaccine. Okay, or sometimes you see it written as TDP vaccine. Okay, so there is the vaccine that helps protect people from diphtheria, tetanus, and pertussis. Okay, so remember those three. Diphtheria, tetanus, and pertussis can be prevented with that vaccine. Okay, so when you think of upper respiratory system infections that are bacterial, that are caused by strep pyogenes, think of pharyngitis, scarlet fever, diphtheria, and that they're all varying in symptoms or severity because of the idea of exotoxins. And remember, we learned that exotoxins are the more lethal of toxins that microbes can produce, because exotoxins, the bacteria is still alive. So it can keep pumping out that toxin into your patient and hurting them. Now, with the upper respiratory system, you can also get viral infections, and the big, most common one is the common cold. And when you think of the common cold, know that they're actually caused by all different viruses. So there's over 200 different viruses. A lot of times you'll see them called rhinoviruses or coronaviruses. And any of you who also are interested in veterinary information or who have ever had pets, coronaviruses are commonly found in dogs. So the dog will end up showing diarrhea and GI issues from coronaviruses. Okay, SARS is also a respiratory coronavirus. Okay. Now, with the common cold, you'll notice that people will be sneezing, their nose will be stuffed up, they'll have that kind of congestion, but they will not have a fever. Okay, so when you see fever, that's more flu-like, whereas common cold, no fever. And with this infection, always remember antibiotics are of no use because it's a virus. Okay, we already learned antibiotics will do nothing for the virus, but what will they do? They'll kill off the normal flora, and they'll end up making your patient much more susceptible to superbugs and to all other kinds of infections. So when your patients are, coming in complaining of something that's clearly just a cold, don't let them bully you into giving them antibiotics just because they want to leave, with a pill that they think will make them better, when it will just make them worse. And what I want to point out, what I like about this slide, is that I threw in the little best foods to fight cold and flu over here. And, so many times we think, oh, that's just an old wives' tale, or, you picture little old grandmas telling you these things, and you kind of like tune it out, like, oh, yeah, sure, that'll really work. And, you sometimes think, well, if it's not, real medication or antibiotic, it won't do anything. But the funny part is that all of the things in this diagram over here, in this little picture, all of these things do actually help in terms of fighting any kind of infection, whether it's the cold or the flu or bacterial or viral, because think about their biological traits of some of the aspects of these foods. So for instance, things like ginger and turmeric, they're vasodilators. And what do you know about vasodilators? They open up your blood flow, they increase blood flow. And why is that good when you have an infection? Think about it. Increased blood flow. Well, now my white blood cells and my immune cells are going much faster, much more robustly to the sites of infection. So these things help your immune system. And things like honey, for instance, they have natural antimicrobial properties. Okay, so for a lot of these things, these will actually help because they're immune boosting, and they help your blood flow, and they help the white blood cells get where they're going. Okay, so, don't believe when someone tries to tell you, place this stone on, your wrist, then you'll feel better. No, those things don't work. But there's a lot of biology behind certain foods and herbs that actually do work and will help you. And notice that in that picture, one of the things is water. Always make sure that you're properly hydrated, especially when you're sick, and that your patients are properly hydrated as well. Okay? Now, that was the upper respiratory system, but as we mentioned, sometimes things get a little worse and patients may end up with lower respiratory system infections. The ones on this list were first focusing on bacterial infections. So whenever you see these terms, keep in mind bacterial, okay, and that they are the lower respiratory system. So be very comfortable with what's an infection of upper versus what's an infection of lower respiratory system. The first one we have on this list is bronchitis. And as you can tell by that word, this is inflammation of the bronchial tubes or your bronchi. Whereas pneumonia, what's infected and inflamed now, that's your lungs. Okay, when you hear pneumonia, that's inflammation of the lungs. The next one on this list is tuberculosis. And anytime that I ask about tuberculosis, I'm going to ask you the same questions. Okay, you'll see this come up on one of the slides in a minute, and you've heard me say this before. When you think of tuberculosis, first ask yourself what causes it, which is mycobacterium. Okay, mycobacterium, which we said are the ones with mycolic acid in their cell wall. And because of that mycolic acid in their cell wall from the mycobacterium, how can you stain for infections like tuberculosis? The zeolniacin acid-fast stain. Okay, so if you can't remember how to spell it, that's okay. Just always know acid-fast stain is for mycobacterium, such as tuberculosis. And ask yourself, can you remember the other one that stain can show you, which is leprosy. Okay, so whenever you hear tuberculosis, think acid fast. And then the last one on this list is pertussis, and the other name you hear that by is whooping cough, because of that cough, that constant cough, which makes that whooping sound. And why is that cough? That's because in that person, their ciliary escalator is destroyed. So that was part of the immune system that we talked about. So their ciliary escalator, which is supposed to help push mucus out of your respiratory system, now that's destroyed, so the mucus is stuck there. And so you reflexively will start coughing to try and get it out, trying to get that mucus out. Okay, so this can be visualized in the next slide. So this slide shows you the idea of whooping cough. And in this little picture down here, okay, right over here, that shows you the pertussis, the Bordetella pertussis bacterium. They're attaching to the cilium, And what they do is they release toxins, and their toxins will destroy these cilia. So now, with that cilia in your respiratory system destroyed, mucus cannot move up and down. So as I was just saying, you end up with that constant cough because the cilia have been destroyed by toxins, and you now have no way to properly get the mucus out of the respiratory system. So you coughin it up trying to get it out. Now, one of the problems with this is it also produces a capsule. So these bacteria really attach to all of those cilium. And that's part of why it causes such a big problem, because they really stick all along that respiratory tract and can release those toxins to destroy those cilia. Okay? Now, the other problem here is when you're dealing with toxins, they can enter the bloodstream. So you are going to see that this will be a more problematic infection than, let's say, some of the upper respiratory problems with, let's say, just having, a sore throat. Okay. Now, as I mentioned before with diphtheria, the good thing with pertussis is that you can prevent it, and ask yourself, how do we prevent it? The DTaP vaccine, okay? So as I said, sometimes you see the letters arranged slightly differently, TDP, DTaP, same thing, one vaccine that helps prevent diphtheria, tetanus, and pertussis, okay? Now, one other thing that I want to mention with pertussis, we said that it's the ciliary escalator that has been destroyed. So just to always keep reminding you of things we talked about in the past, what part of the immune system is the ciliary escalator? That's the innate immune system. Okay, so remember, any of those barriers for infection, that is innate immune system. So now just a brief little slide on tuberculosis. As I mentioned a minute ago, tuberculosis is caused by Mycobacterium. Remember, that is different from Mycoplasma. So Mycoplasma has no cell wall, whereas Mycobacterium, they have a cell wall, and it's very heavy in mycolic acid, which is why what stain can identify it? The acid-fast stain. Okay, Zeal-Neeson acid-fast stain. Now with tuberculosis, kind of picture what symptoms you think of a patient having. So you think of that, coughing up blood symptoms. So remember picturing the red stain for the acid fast shows tuberculosis positive. And interestingly enough, the symptoms of TB, things like coughing up blood, becoming bedridden, becoming very pale from the pooling of the blood, this actually led to people creating the mythology of vampires, because people didn't know what tuberculosis was back in the day. They didn't know that there was this bacteria causing all of these symptoms. All they knew is that suddenly a person would be very weak, have to lay in bed as if the life was draining from them. They would be laying in bed so long that, the blood would pool away from the outer surface of their skin, so they'd appear very pale and drawn. They were coughing up blood all the time, so of course they had the remnants of blood around their mouth, and they would very quickly succumb to this infection, and then once they passed away, their relatives would start to get the same drawn look, pale look, blood around their mouth, and they would get the same kind of, appearance and then pass away. So basically, if you think about vampire mythology, that pale skin, even the sensitivity to the light, people with tuberculosis get photoreceptor sensitivity. So the sensitivity to the light, the idea of that blood and of the highly infectious nature of it, they didn't know bacteria is highly infectious. All they thought was that the deceased was coming back and feeding off the life of their relatives and taking them with them as well. Okay, so it's a little interesting fun fact. Whenever you hear vampires, think of the fact that it was actually tuberculosis. Okay, and I put some other causes on here as well, so it's not always Mycobacterium tuberculosis. Sometimes it's bovis or avium, but with these, notice that in all these cases, it's mycobacteria. Okay, so the end word might change, but the genus of mycobacteria stays the same. Now, in terms of diagnosing tuberculosis, I'm pretty sure that what's on this page you have all experienced, because anyone who goes to volunteer or work in a hospital, you have to get tuberculin skin testing. Okay, so as you can see in this picture on this slide, you look to see if a wheal, so that's WHEAL, wheal, is produced when someone injects a little bit of the purified protein derivative or antigen of tuberculosis underneath your skin. Okay, so you will hear this test called PPD test. Again, that stands for the purified protein derivative, which is a fancy way of saying antigen. They are taking an antigen or proteins from the tuberculosis bacteria, they're putting it under your skin, and they're looking to see whether or not you have the reactions to it. Does your body have antibodies that will instantly fight this reaction or attack this antigen and puff up the skin? Now, what's the problem with this test? Okay, if any of you have had friends or yourself from a foreign country, you know that they will always get a false positive on this exam. On this test, this PPD test, because many people from foreign countries, it was required for them to have a vaccination against TB, because many foreign countries, such as India, TB is so prevalent that they have to use vaccinations for it. And so if you've had that vaccination, which is called the BCG vaccine, so Bacillus calmette-guerin, you don't have to know that terminology, the BCG vaccine in these foreign countries that were given to people, these people now have the antibodies for TB. So they did not have TB, and they do not necessarily currently have TB. But because their body has the antibodies that they produce from the vaccine, they will always show a positive result on a PPD test. Okay. The other problem with this test, as you can kind of pick up from what I was just saying, is this test will show you if your body has antibodies against TB. What doesn't that tell you? Whether or not it's a current infection. You may have a previous infection or may have had a previous infection, and now you'll come up positive from this test because you have the antibodies. So the skin test does not show you whether or not it's a current infection. For things like that, you need to have an X-ray or CT, full-blown, culturing of your sputum for that. Okay, but at least it is a way to kind of initially detect whether or not you've been exposed. They're also developing newer rapid blood tests and PCR testing, which, as you know, PCR testing is nucleic acid-based testing. Okay. First, we have the typical pneumonia, which is pneumococcal pneumonia. Okay, this was one that you see a lot of times in terms of advertisements for the vaccines and everything. With pneumococcal pneumonia, it's caused by strep pneumoniae. Okay, so interestingly enough, you hear strep again, like we did in our labs. Okay, with these, since you already know strep is gram-positive, right? With this one, it's a diplococcus structural arrangement, meaning so it's in pairs. And with this, ask yourself, so if I gram stained the sputum of a pneumonia patient, what would it look like? Well, gram positive, it would be a purple gram stain. And because it's strep-based, strep pneumoniae, it would be round shaped. Okay, so always remind yourself, what would a gram stain of this culture look like? Because there are a lot of cultures that you take in hospitals to gram stain. Okay. Now with this, the patient then ends up with a lot of fluids filling up the lungs and red blood cells, and this kind of uptake in the lungs then interferes with oxygen, proper oxygen maintenance. Okay. Now, like I just mentioned a minute ago, you hear a lot of advertisements for vaccines. So the good part about typical pneumonia is that there is vaccination against it. And it's specifically a conjugated vaccine. So if you look back at our other slides, conjugated simply means that they tie a weak antigen to a strong antigen so that it really produces a much better immune response for you when you get that vaccine versus whether, had they just put one of those antigens into your system at once, because you really want a good immune response when you get the vaccination so that you know that you have proper antibody production. Now, this adorable slide of what looks like little fried eggs in the corner, this is now atypical pneumonia, okay? Atypical pneumonia is caused by mycoplasma, so mycoplasmal pneumonia, and that should sound very familiar to you, okay? So this causes walking pneumonia. It's mycoplasma. We mentioned this guy before. Now, what did we say was significant about mycoplasma? Well, this is the one that has no cell wall. And when you think about the bacterial structure, the idea that it has no cell wall is significant because it means now to target this infection, which is going to be an infection of respiratory issues, low fever, cough, headache, things like that. To target this infection, you cannot use things like penicillins that will target the cell wall because this bacteria doesn't have a cell wall, okay? So what they have to do instead is rely on things like tetracycline. So tetracyclines, they target ribosomes. So when you think of ribosomes being targeted, well, what are they blocking now? Protein synthesis in that bacterium. And as you know, proteins, they do just about everything in cells. So when you mess with proteins, it's a good way to target and destroy something. Okay. Now, interestingly enough, the idea of no cell wall doesn't just affect how you treat it, but it also affects what it looks like. So this little corner picture here, that fried egg appearance is basically that if a cell has no cell wall for a bacterium, then they could take any shape they want, okay? So they could be anything they wanted. They chose to be an egg. They basically tend to concentrate together in the center, and then you see a thinning out of colonies around the edges. So whenever you hear mycoplasmal pneumonia, I want you to think of a few things. I want you to think of atypical walking pneumonia. I want you to think of no cell wall, so penicillin or similar antibiotics won't work. Instead, you need tetracyclines. And I want you to remember fried egg appearance. So whenever you see fried egg appearance, boom, that tells you you're dealing with mycoplasma, which has no cell wall. Okay? Now, the last one that I want to mention for the respiratory system is influenza, which is the flu. Now, keep in mind, if you notice what we talked about in these slides so far, you'll notice that the flu is lower respiratory, whereas the common cold is upper respiratory. Okay? So when you compare these two things, if you think about someone who has a cold versus someone who has the flu? Well, if you have a cold, sure, you're not feeling that great, but odds are you're still feeling, well enough that you could drag yourself out of bed, you could get to work, you could go to school still. Whereas when someone has the flu, what do you notice? The symptoms are much worse. Now they have things like fever, chills, bad headaches, muscle pains. And when you have the flu, you end up calling out sick from work, from school. Okay, so that will kind of help remind you which is which in terms of location, because we said that upper respiratory systems are not as severe, whereas lower respiratory systems, these are now more severe. Okay, now when you think of the flu, the other thing to remember is that it's a RNA genome virus. And as you know, anything with RNA genomes in terms of microbes, they're going to be much more problematic, okay? With RNA, you have gene reassortment, you have prone to mutations, you end up having issues where the high mutation rates of these viruses make it very difficult to treat them. And also even with vaccines, you'll notice flu vaccines, you have to get different ones every year, because by the time the next year comes around, the most common strain has now mutated enough that, the antigens, the antibodies, they're not recognizable at this point, okay? The other problem with the flu that I want you to remember is that the most common complication is going to be bacterial superinfections, okay? And if you ask yourself why, this is something that we've discussed a lot already. You think about patients. When patients come in to see you and they're really, really sickly feeling, what do they want? They want to be given a pill, or they want to be given a medication so that now they're not going to want to leave unless they have that medication. But as you know, what's causing the flu? It's a virus. So if these people are constantly being given antibiotics for the flu, for a virus, for something that, they may not have been cultured at that medical facility, well, now what did those antibiotics do? It killed the normal flora. And so now the pathogens that are resistant, they rise up, they take over, they're now replicating, they're very robust, and now this patient has bacterial infections in addition to the flu that they've trying to fight. Because think about it, once you have the flu, you're now also immunocompromised. So messing around with things like antibiotics, that just makes it much, much worse situation. And now it's time to show a little skin. So this first slide here that we cover, I want you to circle, star, highlight it, because these terms come up not only on the exam, but in all of your other classes, especially nursing-based or medical-based classes, as well as any case studies or information that you will read as you move forward in your career. Now, what we have here are the different terms that you'll encounter for different types of lesions. And I basically put them in order of least gross to most disgusting. Okay, so the first lesion that we have is a macule. You see over here. These are any of the flat, non-palpable lesions, meaning they'll be flat and they won't be raised up, able to be kind of touched. The way palpable means it's raised up, it's touchable. Papule, on the other hand, now is raised up. Okay, so what I like to do in my notes is circle or underline the PA and papule to remind myself that it is palpable. Okay, it's raised up and touchable. Then when you move to a vesicle, now it's not only palpable, but it is fluid filled. And there's another term I want you to remember for vesicles, because you see them a lot in case studies. And that term is bulla, B-U-L-L-A. A bulla is simply when it's a very large vesicle. So basically greater than 1/2 centimeter in size. So sometimes you'll hear infections or diseases referred to as bullous. bullous. And if it's bullous, then it means that it produces lesions that are very large and fluid filled. The last category that you encounter are pustules, and that's similar to a vesicle, except that this one is specifically palpable but pus filled. So instead of saying fluid filled, pustules are pus filled, meaning that they have a mixture of white blood cells, cellular debris, and dead tissue in them. Like I said, the most disgusting. So here's a recap just to remind you all the terminology, and it includes the term bulla, which I said is important to know as well. Wow, will you look at that, bold and red in color. Wow, that would make a great multiple choice question. What we have here is the most common of the skin infections, and that is acne, something that I'm sure a lot of you have had experience at one point or another. I know, we were especially plagued by it in the hormonal younger years. And when it comes to acne, there are a few different things that can cause acne, and most people think of hormonal, clogged pores and oil, which you see on the slide referred to as sebum, as causing acne. But not all forms of acne are simply hormones and clogged up pores. Some of them are actually infections, specifically caused by really long word bacteria, Propionibacterium acnes. And now that's kind of where you see that some people who struggle with acne, they'll try all of those treatments and cleaners and cleansers and exfoliation and all that, and they won't see improvement. And that's because in those cases, it's an infection, it's bacteria. And so what they end up needing is a prescription to kill the bacterium, okay? So just keep in mind when you see acne, it's not always hormones and clogged up pores, that sometimes it's from an actual infection, and it ends up being the most common of the skin infections. So circle that title, put stars next to it. Now when we go through skin infections, you'll see some, bacterial friends of yours that you remember from other lectures and from lab especially. So on this slide, for instance, you see that Staph epidermidis and Staph aureus both end up causing skin infections. And we mentioned with Staph epidermidis that he's normally not one of the bad guys. In fact, 90% of your normal skin, normal flora, is made up of Staph epidermidis. But like we always say, when you think of these things, normal flora especially, they can be opportunistic pathogens. So he can end up causing various infections. You also have the big bad guy from lab, who is Staph aureus. And Staph aureus is not just going to be seen in terms of staph skin infections, but also things like food toxicity as well. Now, with Staph aureus versus Staph epidermidis, you recognize some information from lab, and that's kind of the key point that I want you to put on this slide, is distinguishing these two species of Staphylococcus. Okay, so first of all, you notice this term here, coagulase negative epidermidis versus coagulase positive aureus. So make sure to write on the slide and circle star highlight the fact that Staph aureus is coagulase positive, so you can test it using the coagulase test, specifically by using rabbit plasma. And if it solidifies, boom, that's Staph aureus. Another thing I want you to remind yourself of on this slide and to put stars next to is if your patient is suffering from one of these two infections, Staph epidermidis or Staph aureus, what's an antibiotic that they are both sensitive to? Right away, you should write down novobiacin. Remember, Staph epidermidis and aureus get killed by novobiacin, whereas saprophyticus did not. So if your patient is suffering from a staphylococcal skin infection of epidermidis or aureus, you would want to use novobiocin as one of the options, and they'd both be sensitive to it. The last reminder that I want you to write on this slide is the idea of partially why Staph aureus is so pathogenic. And if you remember hemolysis and what that means, remind yourself that Staph aureus is beta hemolytic. So beta is bad. Chews up or destroys all of the red blood cells. Okay, so produces some nasty toxins and can really do a lot of damage. Now, when we talk about staph skin infections, you're going to see a lot of terminology, kind of like when we just went over lesion terminology. I want you to know the difference amongst them. So first off, you have folliculitis and staph infections. Okay, so folliculitis is specifically when the staph skin infection has targeted the hair follicles, and it becomes referred to as a staph if the hair follicles are specifically those of your eyelashes. So you would only have a staph on your eyes, whereas anything on, let's say, your scalp or even on your hair follicles on your arms or legs, that would be folliculitis. The other terminology difference I want you to be aware of is a furuncle versus a carbuncle, which I know are kind of funny sounding words. With a furuncle, this is a boil, like, I'm sure a lot of you have seen lovely gross pictures of boils. And that's a type of lesion or abscess where you end up with a lot of inflammation and pus. So it kind of looks like in the lesion slides when we were talking about the really gross vesicles and pustules. So now this would be like a pustule. And if that ends up getting very deep and pretty much spreading and to a sepsis level, then it becomes a carbuncle. And there's a trick that I have for remembering that a carbuncle is simply a very deep spreading furuncle. And that is circle or underline car at the beginning of carbuncle. So car, you think of a vehicle, car is traveling. So carbuncle is once the furuncle or boil infection has traveled deep tissue and maybe going septic. Then we have a few different conditions that we kind of list out over on the bottom of this slide, which is impetigo, scalded skin syndrome, and toxic shock syndrome, which you've also heard in lab, and is not only caused by Staph aureus or staph infections, but you'll also see with strep pyogenes infections as well. So when we talk about these infections, notice impetigo says non-bullous sores. So they're not going to be the very large vesicles in that one. But most importantly, it says spread by autoinoculation. This is something I want you to pay close attention to because so many people don't realize that it's actually you who spreads your own infection sometimes. So autoinoculation means that with a lot of your skin infections, you self-spread them from one body part to another. So for instance, if you started off with having a skin infection on your neck, let's say, well, you go to scratch it, and then without washing your hands, now you go to rub your eyes. Well, you just traveled, you spread that infection from your neck to your eyes, okay? People do that all the time with their scratching, their wiping, kind of think of when you wipe sweat off of your forehead and all. Well, now you may be spreading infections that you had on your hand or your arm without realizing it. So that's what autoinoculation means. It means you spread microbes from one part of your body to another part of your own body. The next infection that we mentioned here is scalded skin syndrome. And you have a picture of that on the bottom. And what I want to point out here is the fact that it says bullous impetigo. So this is when regular old impetigo has gotten worse, particularly from toxins being released from the Staph aureus. And with this, now it's bullous, meaning bulla, so the very large version of vesicles. So this will have very large lesions that are fluid-filled. Okay. The last one is toxic shock syndrome, and that one is very sad because we also show it with strep pyogenes as well. And the reason that I say that one is sad is because this is the one where you see people your own age, 19 through 23, if you look up in the news and whatnot, you see they've had amputations because of toxic shock syndrome. So for instance, there are a couple of famous models who are only 19 and 23 years old when they had to have their legs amputated because of toxic shock syndrome from improper use of tampons. So always be very careful, the female students, when you are dealing with anything like tampon usage, because as we go over in medical micro, it's very easy for that to trigger toxic shock, septic, staph infections. Because think about it, for instance, if girls are using super absorbent tampons when it is not a heavy flow, or they're leaving the tampons in too long, well, now in that already delicate, thin tissue area, they are now drying everything out, and then removal or insertion of additional tampons during that time period now makes micro tears in the vaginal area. Plus, you have all of this absorbed blood, which is great food and nutrients for staph bacteria. So they love it. They start replicating, and then you have the basically what we call micro tears that you wouldn't even be able to see. They use that as like a parenteral route entry to get into your body, to get into your bloodstream, and they overgrow. And then once they're traveling in their septic, they can do major damage because, as you know, Staph aureus, strep pyogenes, these are bacteria that release a lot of toxins and can be necrotic. And so those young people ended up having to have amputations from these kind of infections. Something as simple as tampon usage ended up causing it, which is a shame. Now, like I mentioned, similar to staph infections, you also have streptococcal skin infections. And as you saw in lab, one of the big causes of streptococcal skin infections is the big bad guy, strep pyogenes. And so as soon as you see strep skin infections, there's a certain antibiotic that I want you to remember and remind yourself of. So if a patient is suffering with strep pyogenes, what can you treat it with? Bacitracin. Remember, bacitracin eats pie is the trick to remember that. Also, always remember gram staining of these different bacteria. So in the case of strep, you know that it's gram-positive, coccus shaped, and you have that strip of strep, meaning you'll find it in the linear fashion. This slide also reminds you of something else you remember from lab, which is the beta hemolytic aspect. So again, remember, beta is bad, and that's the one that completely destroys red blood cells. And you kind of need your red blood cells, so you don't want beta hemolytic bacteria infecting your body. Okay, so as I just mentioned, one of the big bad guys with regard to streptococcal skin infections is your good old friend strep pyogenes from the lab. And with strep pyogenes, just kind of, we already mentioned it a second ago, but for what do we already know, remind yourself of bacitracin eats pie, so treating with bacitracin for these skin infections. That's why things like triple antibiotic, Neosporin, they tend to have bacitracin in them. Remind yourself what you already know, the gram staining, what they would look like, and remind yourself about the hemolytic activity. Then what I also have on this slide is some examples of the virulence factors, which basically means the various proteins or enzymes within that bacteria that allows it to be so harmful to you, how it does damage. So basically the toxins and enzymes that help it to break down your tissues, destroy your cells, and really stick or stay in your body. So here you see some of the damage that streptococcal skin infections can do, specifically strep pyogenes, and you saw in lab as well some of the even gorier, more dramatic photos. This guy, he can cause the common sore throat, which isn't very bad, but if he's then producing toxins and really going into that hemolytic activity, you end up seeing some very scarring skin infections. And so you see erysipelas, which is the first description here, which is more on the outer layers of the skin, but can also end up septic. And then you have the two that we've mentioned in lab already, which is necrotizing fasciitis and streptococcal toxic shock syndrome. So, like I've kind of mentioned in lab and lecture, with necrotizing fasciitis, the two big culprits of this one are strep pyogenes and then something called Vibrio, which you usually find in marine waters. And you see this can also cause shellfish food poisoning as well, the Vibrio bacteria. But in the case of strep pyogenes, when it ends up causing flesh-eating disease, necrotizing fasciitis, it's mainly due to the exotoxins that it's producing. And what happens is these exotoxins end up serving as what we call a superantigen. So basically ask yourself, when you hear the term antigen, what does it mean? Well, that's the protein name tag that triggers the immune response, right? So if it's a super antigen, then it's triggering a super immune response. And you may say, well, my immune response, that's there to protect me, to help me. Well, think of a really big immune response. What else does that include? That includes a whole lot of inflammation and cell destruction, cell targeting, all this kind of stuff that can actually end up damaging your own cells. So sometimes too much of a good thing ends up being a bad thing. So the superantigen ends up causing your own body to do a lot of damage to itself. The last one here is streptococcal toxic shock syndrome, which is similar to staph toxic shock syndrome. It's very destructive. And I know before I gave the common example of tampon usage causing this, but it's not always that. In the case of streptococcal, especially, for instance, in the news not that long ago, there was a father who caught strep throat from his young son. I think the son was probably only around 8 or 9 years old. And the father's strep throat, because strep pyogenes can end up producing very bad toxins and go septic and everything, the father's version of that strep throat that he caught from taking care of his sick son ended up going into full-blown toxic shock syndrome, and he had to become a quadruple amputee from it. So something as simple as strep throat, sore throat infection, can end up becoming something where this poor man lost all 4 of his limbs. So you always have to keep in mind, be very careful of infections, no matter how mild or unthreatening you think they are, because under the right conditions, sometimes they end up becoming a very big problem. So you may think you're doing good taking care of someone who's sick, but if you're not watching out for your own safety at the same time and taking proper precautions, that kind of like we've gone over in lab, then you can end up picking up something that ends up being very, very damaging. Another lovely one to see is leprosy. And for leprosy, we've mentioned this before with regards to diagnosing it. So remember, with leprosy, because it's caused by a mycobacterium, just like tuberculosis is caused by a mycobacterium, for leprosy, we said that you can use the zeolnisin acid-fast stain. Okay? So for what did we say for leprosy? Make sure to write down that you can use the acid-fast stain to diagnose a culture as being positive for leprosy or tuberculosis, and specifically the red cells. So again, picture the coughing up blood, the blood of losing limbs. The red cells will be positive for mycobacterium, whereas the blue cells will be negative. And there are actually different forms of leprosy. And because it is neurological, it ends up having some paralytic or sensation loss, as well as the loss of limbs and really disfiguring nodules all over the body. You especially see leprosy in third world countries, but we do still have issues in America. Interestingly enough, armadillos are carriers of leprosy, especially down in Texas and southern states. So as cute as they are, never hug or kiss an armadillo. That's the big lesson to take home from microbiology. Now, bacteria aren't the only things that could cause skin infections or skin diseases. Here's a list of a few of the viral skin diseases, and we're going to go one by one to take a quick look at them. So first up, you have measles, and what measles ends up doing is you get this rash that starts in the face, and it progresses to the really bloodshot red eyes and a really bad cough. Now, a lot of people, when they think of measles, they also associate it with mumps and rubella because the vaccination that has now prevented measles from being the big bad guy that it used to be also covers vaccinating you for mumps, which is the swelling above the jaw, specifically the parotid gland or your salivary glands. And it also protects you from rubella, which is a face rash that you end up having for about 2 to 3 days. Now, with measles infection, the big problem with it is that 1 in 1,000 cases triggers encephalitis, and in some cases it's called subacute sclerosing panencephalitis. But either way, anything to do with encephalitis becomes a big problem, as you're going to see at the end of today's lecture, because anything that involves the brain or the nervous system can be very, very damaging. And since measles was especially big at targeting children, you definitely don't want anything in a child causing swelling of the brain or neurological damage. But again, thankfully, there's now the vaccine, which has helped really dramatically reduce the number of cases of measles. But at the same time, unfortunately, due to those anti-vaxxers who think that, surfing the internet for 10 minutes in their basement makes them scientists and doctors and medical experts, problems like measles are starting to come back up because people are not doing the vaccinations that they're supposed to, and that's now causing a problem with not having as much of a herd immunity like we should. Another viral skin infection, which we actually mentioned once before in this class, is smallpox. And smallpox is caused by pox viruses. And what did we say about pox viruses? Well, they're the ones that have a DNA genome. Okay, so remember they are DNA genome, and they're the largest animal virus. Okay, so for pox viruses, we already said DNA genome and largest animal virus. Now, smallpox is another one that used to be a very big problem and had even affected the respiratory system and could be septic in terms of traveling through the bloodstream. But the good thing, again, about smallpox, like with measles, is the number of cases has dramatically decreased because of vaccination. So the introduction of the smallpox vaccine has predominantly eradicated this. Now, it is still in the news every once in a while as a potential bioterrorist agent. And also keep in mind that a lot of these big bad guy viruses and bacteria, they are kept or housed in small quantities at various locations for research purposes and to basically try and protect the public. But, things like that can always end up in the wrong hands. So it does pop back up in the news every once in a while. But again, the vaccination aspect has really helped protect us from it. Then there is chickenpox, which you are all pros and experts at by now because we've mentioned it before with regard to what is its genome. So chickenpox, when you hear that, genome is DNA, okay, because it was one of those on this slide that had DNA viruses. You also know that it's actually a herpes virus, not a pox virus, okay? Make sure you put a little star in this slide reminding yourself that herpes virus is a herpes virus, not a pox virus. That's very important because when you think of herpes viruses, it reminds you that chickenpox is one of the ones that can go latent. And you have to know that if it goes latent, what does it come back as? We'll see on the next slide, shingles, which is very painful because when viruses like chickenpox, causative agent, go latent in your body or dormant, they tend to do so in nerve cells. So if they're going dormant in nerve cells and then popping back up, reactivating lysing cells, it ends up being very painful. Now I want you to think of when would this be most likely to happen? When would latent infections tend to reactivate? The two big examples are one, when you are under a lot of stress, and two, when you are immunocompromised. Okay, so those are the two times that latent infections tend to pop back up. One other thing that I want you to circle, star, highlight, I've bolded it in red, is the fact that chickenpox has been associated with Reye's syndrome. So, back in the day, they used to think, oh, chickenpox, my kid has chickenpox, I want them to be immune to it. Let the kids play together, let them hug, kiss, whatever. I'd rather my kid get chickenpox now rather than older. Turns out there are two big problems with that. One, if your kid then ends up during the chickenpox infection with Reye's syndrome, which is usually increased by usage of aspirin as parents try to, reduce fever and whatnot. Reye's syndrome ends up causing brain dysfunction, which you do not want in your child. Secondly, like we just said, once your child has been introduced to chickenpox, now they have the potential to have shingles at any point in their life, which is much more damaging and painful than the initial chickenpox infection because, again, this is now once it's been in the nerve ganglia. The good thing is they have developed a vaccine for it, and if you get the vaccine, then you won't have to worry about chickenpox or shingles down the line. So as we just mentioned on the previous slide, the idea with chickenpox that I want you to circle, star, highlight is that it does reactivate later in life as shingles. And what's really interesting is depending on which nerves it goes dormant in, ends up producing different patches or locations on your body. So for instance, as you can see here, it ends up only being half of the body that demonstrates the big painful lesions and skin disruptions. Now, like I mentioned on the previous slide, keep in mind that stress and being immunocompromised are the two big factors of when this will reactivate. Okay, now if you had the vaccine instead of being younger and having chickenpox itself, then you don't have to worry in terms of shingles popping up. Okay, and like I said before, this ends up being much more brutal and painful because of the fact that the infection went into the nerve ganglia. Please don't forget to head over to part 2 of this week's video lessons, my personal favorite since it traumatizes the students the most.